Alright, in this recording we want to specifically look at phage, or viruses that infect prokaryotes bacteria. So here's our great slide, a picture of a prokaryotic cell being attacked by phage, those little yellow lunar landers injecting their DNA into the cell. Now, what we want to think about is there are two phases to a bacterial viral infection. And the first one is more active. It's called the lytic cycle. So this is when a phage is virulent. It's going to literally infect the cell, make more virus, and destroy the cell as it is releasing more virus from it. Now, the most primitive of all kind of immune systems that we've seen are seen in prokaryotes, and these are restriction enzymes. So these are enzymes that the bacteria will harbor that can recognize specific sequences of DNA and chop them up. So when the virus injects its DNA into the cytoplasm, the hope is that these enzymes would chop up the DNA before they have a chance to produce viral proteins, take over the cell, and destroy it. This is the source of the tools, the restriction enzymes that we will use in the lab to cut up and manipulate DNA via biotechnology. But let's take a look at the lytic cycle first, and then we'll compare and contrast that to what's called the Lysogenic cycle, where the viral DNA incorporates itself into the bacterial genome. The lytic cycle, though, is our first virulent life cycle. We're going to see the virus attached to the cell, inject its DNA, and the virus can also be very aggressive. It can be on the attack as well, trying to degrade the host DNA. So if it's successful at that, then it can take over the machinery that remains in the living cell and make more viral DNA as well as proteins, which then can be assembled spontaneously into virus particles. So in essence, when I say spontaneously, if I took all these pieces into a beaker and shook it up, out the other end would come these phage assembled. All right? So that's the way they're built. Now, ultimately then, we see a release of these viruses destroying the cell so that they can go on and infect a different cell. So that is, again, the lytic cycle. We're lysing the cell to break it open as to lyse something. But we want to contrast that then with what's called the lysogenic cycle. And this is where things get very, very interesting when it comes to viruses. So the lysogenic cycle is when the invading virus is going to incorporate itself into the host genome. So the DNA of the virus will actually become part of the DNA of the host cell. All right? In this state, a prokaryotic virus, a phage, will be known as a prophage. So if I'm a prophage, or if I'm a phage and I've incorporated my DNA into the host cell's DNA, I may have damaged the cell, but in many cases I haven't. Now it happens. As the cell divides, it's going to copy my DNA right along with it because now I'm part of the host genome. This happens to us as well. Now, a couple more pieces of vocabulary now as we talk about this. So what we can contrast now is the lytic cycle, where phage is actively being produced, versus the lysogenic cycle, where we integrate the phage DNA into the host genome. So a prophage is the integrated viral DNA of a phage in the bacterial cell. And we're going to contrast that with what's called a provirus. And that's when we see it in an animal cell infection. So when we have a retrovirus, which we'll talk about, that's a virus that has RNA as its genome. As it reverse transcribes the RNA to DNA, and that DNA is incorporated into, for example, your cells, is known as a provirus. And you have proviruses in your cells. Right? They're there, and every time your cell divides and reproduces, pardon me, it makes a copy of that virus right along with it. So if we look at both of these life cycles, we see that when a phage infects a cell, it has two options. It can either go lysogenic or it can go lytic. And to dictate which direction it goes, we have very mechanical mechanisms. Okay, we have a method based on environmental input, ultimately, to the cell to dictate whether or not a prophage is going to hop out and go lytic on you. So let's look at what's called the operator region for the example phage here is lambda phage. And so this is now a prophage that has been incorporated into the genome of a bacterial cell. And so what you can see is 3 operator regions. So we know how operators work. If you have a protein binding to an operator, you can block transcription of certain genes. And there are 2 genes that are very important for the control of lytic versus lysogenic, and those are CRO and repressor. When repressor is expressed, the virus will remain lysogenic. Okay, it will repress the virulence of the virus. Whereas when CRO is expressed, now we go lytic. Everything's ramped up, we make more virus, the cell dies. All right? So let's look at this mechanism. So repressor and CRO proteins bind to the DNA in the operator region. So repressor is the name of a protein. It binds to operator region 2 and 1, and it represses transcription of CRO. So it represses, you want to think about it as repressing the virulence. So the bacteria look fine and healthy, but what it's doing is repressing the expression of the CRO gene. It also acts as an activator protein for its own transcription, which is in the opposite direction. So only when you have these very kind of compact, very interesting setups for gene expression, as you see in viruses, can you achieve something, this simplistic mechanism that can make a decision for you. Now, when CRO is expressed, it actually binds to the DNA and acts as a repressor of repressor. So repressor is the protein. I'm repressing repressor, so it's kind of tricky, but you'll be okay if you keep thinking about it. Let's look at the pictures. So lysogenic or lytic. So what we see at the top is repressor protein being expressed, and you can see it binding then to operator region 2 and 1, actively loading RNA polymerase on, expressing more repressor protein. This is effectively going to maintain the virus as a prophage in the lysogenic state. Now, if we get some switch, some balance, if it was switching the balance, and now all of a sudden we have CRO around, CRO is actually going to bind to operator region 3 and block the transcription of repressor protein and promote then the transcription of CRO, and that's going to lead to a lytic cycle. So what causes the balance to shift? So it could seem that once you got repressor going, you're locked in. Well, in fact, it's environmental information, and it makes a whole lot of sense. So UV light can cause a different protein that's inherent to the bacterial cells called REC-A. It deals with DNA repair. And when UV light, it can cause REC-A protein to change from a DNA binding protein to having a protease activity. So proteases are proteins that chop up other proteins. And REC-A then will cut up repressor. That will reduce its binding affinity, and all of a sudden now CRO has a chance. Because if we're not repressing the CRO gene, we get some CRO going. CRO is going to bind and repress repressor, and then we got more CRO protein, and now we're headed for a lytic cycle. So you might say, well, why would we want that to happen? Well, if you get excess of UV light, you're often damaging DNA. If too much UV light can cause the DNA to be damaged irreparably. Now, this is a sinking ship. If the DNA in the bacteria is so damaged that it can't reproduce, you, as a prophage, are done and you're going down with the ship. But if you can abandon ship and be like, whoa, this is an indicator that it's damaged DNA, we need to get out of here, let's go lytic, boom, I make a bunch of copies of myself, and then I throw those out into the environment with the hope of finding another cell to reproduce. Very interesting stuff. You can get down even to the level of how the CRO protein binds the DNA. And this is a recurring theme that we're going to see again and come back to. And this is the helix turn helix motif. So this is literally getting down to the fact that how does this machine actually bind into the major groove, recognize a sequence, and then block the transcription of repressor. So much to learn. Think about this, and we'll talk more.